Matter of active research.
Okay. I thought it was just driven by like wave trains getting merged and
There's
like
diffractive focusing, focusing by currents, nonlinear effects, constructive interference of elementary waves, wind wave interactions.
How do I do that?
Thermal expansion.
I stand corrected. Thank you very much.
All sorts of stuff. Peyton, that can be what you study. You can solve it.
Not enough biology though.
You can do some models with your computer background.
A 2012 study confirmed the existence of oceanic rogue holes, the inverse of rogue waves, where the depth of the hole can reach more than twice the significant wave height.
Are we talking about holes?
Also known as Lily's worst nightmare.
Hey guys, let's talk about rogue holes for a second.
Rogue holes.
Rogue holes. Yeah, it is premature to state. Okay.
47 meters along this transect so far.
Man, fish,
so
where are you?
You see our numbers going down.
Man,
so we're almost at
fish.
the
There's been a lot of coral, but no fish.
So you can go ahead and press it now.
One swam by an
I
Argus,
think I see a fish right there, the blue one, at the bottom
Where?
of the screen, at the bottom of the screen. One fish. We
One
just
fish.
got out of view.
I only see coral and Chef Boyardee sea stars.
Those sea stars really do something for,
X, Y.
what is it, like the names of, it's that marketing.
Oh, viral marketing?
Yeah. Wait, what did you say?
Oh, viral marketing?
No, not viral marketing, but when you like
Name association?
paid placement or something like that. Sounds like it.
Oh, so
We're just like,
product
Brian,
placement.
are you or have you ever been sponsored by Chef Boyardee?
you're just like ravioli stars.
Only by a
Chef
shoe
Boyardee.
company.
By a shoe company?
Really?
Yep.
What shoe company?
Brooks.
Ah.
I had a sponsorship with Brooks for 10 years.
Whoa.
Why?
Are you still wearing Brooks?
I am.
Wow.
Best shoes out there.
Me neither. We're gonna have to look at your shoes later.
There's a fish.
Oh, where?
Where?
Bottom left.
Oh, yep, about to just went out of
Yeah,
frame.
I saw it.
You got a
Well
bait?
done.
Nice. How far are we, Lily?
We are
funny.
60 meters in.
Shoes are just
Nice.
cushiony holes. You can put your feet in.
What do you think
They
about
are.
it?
Cushiony
That's
holes.
really what they are.
Hey, Dr. Megan's tuning in.
Hi, Dr. Megan.
Peyton, you're in trouble. You gave her your head cold.
Oh no.
She's coming. She's coming for you.
Did she say she got sick too?
Yep.
Oh, I'm sorry.
Fish? No?
Is that a
No.
Is that a
Worm?
fish?
I don't know.
Siphonophore?
Siphonophore? So many things.
It's all of the things. So great question coming in. This isn't what we're doing right now, really a fish density study. This is just us trying to use this to better understand what creatures are here. Fish. And we've taken all sorts of other samples of all sorts of other different organisms. But this isn't just kind of a fish-specific study. But viewers, feel free, keep asking those questions.
We like them. They keep us entertained.
Yeah, Dr. Megan, we hope Heather is still watching now, even though you're not on still.
And then, oh, sorry.
You're right there?
No, I just had a thought and then like
It left
paused
you.
midway through. Did Megan say she was back home in Seattle or?
She did not specify.
Oh, all right. Good to hear from you again.
Some more corals coming up.
Yeah.
We have 25 meters left.
Very pretty. I'm gonna take a screen grab. I feel like I can see a lot of brittle star arms sticking out.
For sure.
So for our viewer who just sent us the message in Vietnamese, we are off of Tau in American Samoa, the National Marine Sanctuary of American Samoa.
I thought you translated that.
Yeah, are you gonna try to translate it, Brian?
Oh no, that's what Google was for.
Ah, nice.
What did they say?
They asked where we were.
Oh, okay.
Yeah, I would try to say it, but I would probably horribly offend all our Vietnamese viewers and speakers, because I have no idea. Thanks for tuning in.
10 more meters.
Nice.
What's the next mission if I choose to accept? Which I will.
Photos.
Keep on keeping on.
Yeah.
Hey, a viewer says we were trending on Reddit a few days ago.
We were.
Ooh.
Let me-
That was
No.
the
Hermit
squid,
crab?
right?
We're famous, you
Squid
guys.
of the hermit crab?
Hermit crab.
Wait, was it the hermit
The
crab?
hermit crab was
Was it?
trending.
I don't know.
Scott was trending.
I know the squid was on
Scott
Reddit.
Summer.
Did they say what subreddit?
Well, I know the squid
You wanna
was
do
on
it?
Peyton just went up
Yeah.
to like
Fish!
a 12.
Fish! Hey! Fish!
Well done.
Fish.
Perfect timing, fish.
Perfect timing. We
Yeah.
have 7 meters left.
Yeah.
Way to go.
Nice.
Dr. Megan is at the Charlotte Air Museum.
Oh, is that
Oh,
another fish?
I think so.
At
There's-
6?
Oh,
Oh,
and
there's two.
blue. Yeah.
Where?
1 and 2
Ooh.
at the blue at the
Bottom of the screen.
Oh, yep.
Wait, Brian, did they
There's
say
another
what
one?
subreddit
Oh, wait,
we
that
were-
might be a...
Peyton, they
Trending
have
on?
not.
Okay, I really want to find out that.
I can tell.
Well, Brian, I know the
How
squid.
many meters do we have left?
Peyton.
What?
Lily?
The squid video was
4
on our slash
meters.
videos.
Okay.
Oh, is the piglet squid?
Yeah, that was trending.
Maybe we'll try to do a photo grid.
Yeah.
I
in
really
this
want
area.
the.
There's a lot of stuff here, so
There is. Wow.
if you wouldn't mind
Oh yeah.
marking this area.
No problem.
Awesome, thank you.
Fish.
Another
Oh,
one?
right there at the bottom.
All right, we've arrived.
What is this coily thing on the left?
There is
Above
What?
this
We'll just
urchin.
Get the
It's like-
size of the box.
Oh,
Oh,
Curly.
I'm gonna guess a whip coral,
yeah.
but it could also
see
be
where-
trash.
And that's our
Okay,
survey.
let's see
Awesome.
where she wants to
Okay,
How many fish did we get in our
survey
survey?
is done.
How many fish total?
We had
Okay.
1, 2, 3, 4, 5, 6,
Okay.
because in one shot we had 2. So 6 total.
And 430 meters and at 220.
The viewer
So-
says it was the squid.
It was
Okay.
the squid?
Yes.
Okay.
Mm,
I
that's what I
really
thought.
want the crab.
0220
to get
was
up there.
end time?
0220, yeah.
Okay, awesome. Thank you.
Wait, which squid?
From a previous cruise.
Yep.
Oh, a previous cruise one.
Yeah,
Okay.
from.
Yeah, I don't think the crab
Bear.
highlight video's out yet.
Oh,
So-
someone did put up the self-sampling crab video.
What?
Yeah, they beat us to it.
Oh, that's awesome.
Right.
So,
So 10,
Hannah, do you
1,
want to go
10,
back those
1,
25 meters,
10,
or
I
does
think,
this area look
yeah,
fine?
I think this area is good if
Okay.
Summer's down to do a mosaic.
Yeah, she is.
Yeah.
Awesome.
She's just learning how to do it right now.
No worries.
Third
yeah,
person
where do
summer
you,
is
do you
even
have a
better.
particular place you want to start?
Let's see. Just wherever there's like a more concentrated cluster. Yeah, let's turn a little. Keep turning. Let's see.
Oh yeah, that's fast.
Hey, Jamaica, thanks for tuning
Maybe
in.
go back to starboard.
Is that where it was? Back to starboard?
Yeah, just if you can turn that way just to kind of see.
You got a good overview in Argus Cam too.
Oh yeah, look at that. Okay. Let's try to do this right in front of us. So kind of the cluster to the right of the lasers and then over to the left, just like that small area. It shouldn't be too big. Just do like a, I guess it would kind of be more like a rectangle.
Okay. It'll be easier for us to start upslope.
Okay.
And then step forward and then starboard and come downslope on our grid.
Okay.
Okay.
Yeah, whatever is easiest for you guys.
Alright,
no
so
problem.
I'll lateral over and then we can do this.
Yep, exactly.
Back row, did we ever settle on what large silverfish that was?
And you can kind of gauge like how far out that thing is, and we want to make sure we get way past it.
I think we
on
had
the
some
mosaic.
ones that looked
So
kind
that
of
looks like
similar, but we weren't
5
super positive.
meters might cover that.
I think they're closer.
Let us know when you guys are going to start the mosaic.
Yeah,
5 meters cover
Barrack
that.
Splendid
5 meters ahead.
was
so
one
we're gonna, this is our
thought,
start point.
but it has a different sort of color pattern.
Oh, you're saying that's 5
Because
meters?
it's a little more red
Yeah.
than the
If we
fish we
go
saw.
5 meters forward.
I think this head is a little bit different.
Let us know when you guys are starting, please. Okay. Peyton, can you note start time, please?
Yeah.
They're,
5.
they haven't started
So
yet.
we want to do like, do we want to do 10 and go?
But they're soon to start.
So viewers, we're going to do a photo mosaic soon. So we'll be kind of traveling a zigzag pattern over the area. And if you are looking in Argus Cam, you'll be seeing Hercules taking some lots and lots and lots of pictures. And that's
There's a
something
little
we'll be able to-
jellyfish floating in and out of Argus Cam right now.
It's so cute.
Oh.
We'll be able to create a 3D model
There it is.
eventually
Yeah.
of the area.
So were these the same corals from earlier?
We have, there looks like we have some gorgonians as well, which are soft corals. Looks like we got a combo.
Nice.
Yeah, so hopefully we can capture these in the photo grid that we're about to do.
We're just getting our start point dialed in, and we're going to begin here pretty soon.
Awesome.
Perfect.
And Lily, you got the cameras ready?
Got all those
give me a
little
minute.
creatures up in
Someone
the
else took over a
top
computer
left-hand
I
corner.
need.
Okay.
And Peyton, obviously this
Yeah,
is
step forward another
PM1.
meter, maybe.
Are they shrimp?
They don't seem to be swimming like shrimp.
Yeah, zoom in.
Okay, Peyton, I'm starting the new survey ID.
There's a jelly again on Argus Cam.
Can you go wide video?
I think that's a good start point there.
Yeah, I think so.
Yeah.
So
Kind of also looking at Argus view.
Right.
Yeah.
We're starting, right, guys?
Wait, no.
Oh, okay.
Sorry, Peyton, I'm gonna have to redo the survey ID. Sorry, not sorry.
All good.
I want it to work.
Me too. Do what you gotta do.
Oh, goodness. Okay. It's flashing, right?
Doesn't look like it currently.
Oh, I just stopped it.
Oh, okay.
Okay, no. Okay, cameras are ready, ship's in position. I'm all good.
I'm ready as well. All right, we're gonna start. Ready, Peyton?
Ready?
Starting now. Go.
All right.
The summer has
425
started.
meters depth, start time 0226.
425.
So Lily, any idea in a photo mosaic how many pictures total it ends up taking?
Oh my gosh, goodness, that
depends on the size.
Yeah, that'll depend on the size. So essentially, hey Anthony, is the camera driver up right now?
Yeah.
So if you look at this as we're driving along, we want to get every single object in at least 3, if not 4 cameras, images taken. So like we just passed over an urchin, it'll come up, it'll be bright red in a second. And we want to make sure that's in 3 images. So depending on the size of the survey, we'll have any number of hundreds of images.
Okay, step a meter to the starboard.
Ones we've done, say like a 2x2 meter is about 400 photos, something about that. Because you got about 70–80% overlap in every photo when you're taking them.
Yeah.
So you can extrapolate from there.
So it's
For there
important
to be
to go
enough
really slowly.
points.
Yeah.
What?
It's important to go really slowly, though.
Yes. So our
Very.
ideal height, the camera's focal point is about 3.5 meters. So Herc is at an altitude of 3 meters right now, 4 meters or so. And the ideal speed is around 0.2 knots. But that'll vary based on currents and stuff. So we just want to make sure as we're like going through the pictures that we hit enough pictures of each location. So we're watching these cameras, making sure that points of interest are able to be followed from picture to picture, because when the mosaics formed, they're overlapped, and the software that we created finds all these points of interest, and it essentially locks them in place and creates a spiderweb grid of all the images and forces them all together to make the mosaic.
Oh, a good explanation.
Do you end up with a 3D map of the entire surface, or just objects on the surface that you care about?
We can get one of the entire surface. So
Okay.
Essentially, we have 2 cameras. We have the camera A and camera B up. If you're watching
On the quad view on the lower left.
there you go. And by having 2 different cameras, we are able to get measurements, and by comparing the 2 different depths of view, it's like having one eye. You can't get a distance or a depth. You have no depth perception. But having 2 different cameras, we're able to get different heights, depths, and measurements, which allows us to get a 3D grid and make a 3D surface. So as long as everything's in focus and we have enough overlap, we are able to get the entire surface 3D mapped.
It doesn't look like we're getting that.
Actually, for the people on the ship downstairs in the hallway and I think it's the tween deck, they have a bunch of Roman lab posters, and there's one with all the bottles. They're like
There's
old
the big
water
one of the
jugs.
ship.
There's
too.
one of a ship.
I think that was in the Gulf.
I think these are online too, but I'm not exactly sure where, so I can't direct our viewers. But they come out really cool, and you're able to actually see dimensions because of the 2 different cameras.
Yeah, there it is. Lily, can you zoom in on NavG?
Yep. I'm taking this down for a sec.
Roger.
Yeah, cool, thank you.
No problem.
All right, I think that, yeah, it got it. See the distance on our lines?
Yeah,
So I don't know what happened there.
me either.
It's fine. We
Stuck.
caught it, you know?
Huh.
Wonder if we lost a beam or something.
Yeah.
Yeah, do 5 and see what it does.
Hercules to scale.
Argus not.
Argus not.
We're gonna need a bigger crane.
How long is this grid? It looks like a good size.
The steps that we're doing forward and backward are 5 meters.
Awesome.
And then we're doing a meter over.
Cool.
So
Yeah, that 5 looked
Perfect.
real.
Yeah, looks good.
I buy that. Yeah.
yeah.
So thanks for joining us on this dive. We are currently doing a photo grid of this area. Has a lot of corals that are of interest to us, so we are using the software and hardware from the Roman lab that is on Hercules right now to take lots of pictures, and ultimately in post-processing those pictures will be stitched together to create a 3D model, as Lily so awesomely explained earlier. Now Lily, I have a question. If there were things such as like holes on the seafloor,
I love how you keep drilling into this.
Rogue holes.
There was a rogue hole on the seafloor.
I don't think rogue holes are on the seafloor.
Well, they are
Surface.
rogue.
Are they caused
How
by
do
the
you
seafloor?
know?
I don't know. That's a question for Peyton.
What?
How are rogue holes formed?
Probably the same thing that
Oh,
makes rogue waves, just inverse, so you just have a rogue hole.
okay. Well, if there was hole, as I was trying to describe earlier, the way light works, when it shines into a hole, it actually lights up the hole, which is a crazy concept. And that you guys were just not understanding as I explained it. So it wouldn't be just a black void. That's not, we don't have black holes at the bottom of the ocean. It's not how it works. Sorry to break it to you. So.
Sounds like I'm going to get my next master's on holes.
worth it.
Hole
Holes on the
studies.
seafloor.
Do you
Did a
think
whole
these
study of
shrimp
that.
are
Whole
attracted
study of it.
are just here, or are they attracted to our lights?
What was your question?
What?
There's a bunch of shrimp swimming by Hercam.
Really?
I
Oh.
think that they're
Yeah, look.
probably mysid shrimp or
Maybe they're
opossum
looking
shrimp.
for the darkness of a hole, maybe?
Like I was just curious
They
if
emerged
they
from
were
a
already
hole.
here in this like quantity, or if they were just attracted to Herc's lights.
I think they're feeding and they're attracted to the light because they can, either they see the food or the food is being attracted to the light, which is then attracting them to the light.
So I got some questions coming in about
Having a
ROV
little party.
Hercules and Argus, and I'll start off with Hercules first. Hercules can go to a depth of 4,000 meters, which is just over 13,000 feet. It has a 30 to 45 meter tether, and is about just about 13 feet long, a little under, just over 6 feet wide, and just over 7 feet tall. It has a mass of 5,500 pounds in the air, and it has a max speed of about 1.5 knots. Max bottom speed, or on bottom transit speed, is about 1 knot, and that's without us doing any sampling or anything. And we can ascend or descend at about 30 meters a minute.
You said Herc weighs 55,000 pounds?
5,500.
5,500, okay.
5,500.
Yeah, wow.
And Argus, and you can see all this information and more on nautiluslive.org by clicking on the tech. It can go up to 6,000 meters deep, which is just under 20,000 feet. We currently limit to about 4,000 meters because of tether, because it has a 4,500 meter long cable. It's about 3.8 meters long, 1.2 meters wide, and 3/10 meters high. It has a weight of about 4,700 pounds in air, 3,000 in water, and it can move about 2 knots, and does pretty much the same as Hercules. Takes about 20 to 30 meters a minute ascent and descent rate. But there's tons of more information about all of them I won't list all the specs to you. If you go to nautiluslive.org, if you go to the Tech tab up at the top, you can learn more about the EV Nautilus, Hercules, Argus, our multi-beam acoustic systems, and telepresence. A lot of great stuff there. I encourage you to check it out. And if you're an educator, you can also click on Education.
It's glassy.
Create a free login and enjoy a lot of high-quality free educational resources that you can involve with your students, or if you're a parent, you can do it at home with your kids. We encourage you to check them out, all free. So the Mungo arm, great question as well. That is kind of our blunt force trauma type arm, I guess you could say. It's incredibly strong, but you can't do the same type of specific tools. You're not going to play operation with it like you might try to do with with the Predator arm.
Hey, Hannah.
Yep.
I think we got like that cluster on the right as well as what we just started with. So do you want to keep going starboard? I didn't see much else there.
No, I think that's good. So that total is that 5 by 5 then? The whole grid? About
5 by 6.
Yeah.
Okay. Yeah, that's good. We can stop there.
Cool.
I think we got that cluster that was there. So okay, we're gonna jot down the end time. Thank you very much.
Nice job, Summer.
Yeah.
Yeah, Summer.
Go Summer.
Nice
Go
work.
Summer.
Grasshopper has risen.
You can become the master if you can grab this fly with the Predator arm.
All right, so let's see. Let me look at the map. So yeah, we'll just continue the
W3?
Yep, waypoint 3.
Northeast.
Mm-hmm.
That was
Yep.
alrighty. I'll get the ship moving.
Cool. Awesome. Thank
Nice.
you. Now it looks like there's not much. So that was a 5 by 6.
Every time you ask, they're doing what, 0.2 or 0.3, or is it just the speed of the ship? Oh, okay.
There's definitely not as much here on this dive as there was the last dive.
What's all that stuff
Yes.
over to the right? Is that a rocky outcrop over there?
Is there?
It's just different
Doppler?
colors.
Sure, go for it.
No, it doesn't look like there's any
Just
rock.
different colors.
There's so much
dirt.
sand.
Are you looking at this? Oh, yeah.
We found it. Dirt.
Oh, look at all the shrimp or whatever you said.
What
Kate.
if we tried to sample 5 of them?
You want to suck 5 of those up?
We forgot the net.
Exactly. Where's the butterfly net?
Oh, there's a shrimp on the 6 o'clock on the screen.
It costs extra for the net. It's DLC.
Could we reach up high enough to suck up those shrimp? Are they not big
What
enough?
is all that stuff? To the right?
Oh, what's that to the right?
There's 5 up there.
There's also
To the
something
left.
red to the
I think there
left,
are
too.
5 up there.
There's a
Oh.
large red thing on the
Yeah,
left.
Oh yeah, what
I
is the
think
large
it's a
red?
sea
It's,
star.
yeah, yeah.
Maybe we check out the sea star first. This
Okay.
looks a little closer. Oh.
Accurate.
How many arms is that?
Ooh, looks like
I
1,
see
2,
3,
6.
4, 5.
6, 7.
Looks like one's
Missing 1 or
regenerating.
2.
Looks like 3 of them got nipped a little bit.
Yeah.
You want to
You
zoom
can see
in?
like some of the tips are
5
lighter.
plus 3
what?
nubs.
Oh, yeah.
Oh
And it
yeah,
did
he's growing
not-
an arm back there. Look at that.
Did not win whatever fight it
Can we
got
get
in.
some grabs as
No.
well?
Yeah, yeah. Thank you.
Here, it's in focus now,
Oh,
Peyton.
wow, look at all those.
Thank you. Okay, that's, yes, 8 arms.
There's a little tiny one.
Oh, there's a 9th.
Oh, you're
Oh,
right. Itty bitty
good
one.
eye.
Oh, there's a little brittle star on the left.
What is that?
Brittle stars everywhere. I think that would be
Yeah.
our best chance to get 5.
Mm-hmm. I think we got one with the hard coral sample.
Yeah, but I think they have to be the same species, don't they?
That's
Yeah.
true.
Go with
Oh,
this one?
is that a whip
Yes.
coral?
Yep.
Oh yeah. Can we get a zoom
Just
in
above
on-
it? Yeah, if
Mhm.
you, can you angle the camera up, Summer?
Yes, I can.
Perfect.
Beautiful. Summer's the
Look at
pro.
that.
Ooh.
It's another little scallop at the bottom right of that frame right now.
Oh, hey.
We are missing 2 more scallops.
We're
But they have to
quite
be
a
near each other,
distance
right?
from-
That's true.
I don't think that's a scallop. I think that's a holdfast.
I can't really tell.
You don't need to move quite yet.
I don't think it's a scallop.
Oh.
There is a very tiny red crab and or shrimp to
Oh,
the right of it.
wow.
Oh.
That's tiny.
Hello,
Where?
friend.
And a
Can you
fish.
zoom in
Bye,
more?
fish.
Oh,
I see a
wow,
brittle
yeah.
star. I
There's a
think.
crab in the top right, too. Very top right.
Oh yeah.
Oh.
Hey, buddy.
Right.
Look at that.
There's a lot going on right here.
Mm-hmm.
A lot of little things going
More
on.
corals.
If only there were 5 little things
What's
going
underneath the
5
sea
of the
star?
same little things.
on. Right
white
next to
thing?
each other. That
Oh,
can't
What
run
is that
away.
white
Is
thing
he
under the sea
eating?
star? Is he eating?
Maybe
Huh.
he's
You can
eating.
angle back in on him, so.
Because
Yeah.
it's under the smallest, like,
Beautiful.
arm.
Hmm.
It does look like it's a part of an urchin.
Yeah.
That's
It
the
looks
test.
fuzzy.
Oh.
Do sea stars eat urchins?
I
I don't think so.
I'm not sure.
I'm not sure either, but I don't think they do. I've never seen them.
That looks like a test.
Can we get some good grabs of this?
Yeah.
It does.
It might be a sea cucumber, maybe,
So,
that is just like passing over uncomfortably.
I think this has 10 arms.
10?
Yeah, because in between the long one on the left, the orange one that's regenerated, and the little nub above that, there's a very small one growing.
I thought I saw that really small one already, though.
so
But
that might be a Coronaster helicepus.
So we're going to go
Definitely.
to the right.
I'm
Yeah,
gonna
if
say
we could
Coronaster
see.
species.
Yeah.
There's another whip coral.
Some gorgonians.
There's a shadow like 3 o'clock or 3 o'clock of the lasers that kind of looks like a llama.
A what?
Like now it's right above the
so it's
lasers.
just
It kind of looks like a llama.
directly on, but sometimes
Not anymore, but
Can we zoom in on the gorgonian just to the right of the lasers?
Yeah. Summer, am
The
I
one-
good to zoom right now?
primnoids.
The one right below the lasers. The one
the
right
one just
below
under the
Just
lasers.
right below the lasers.
You good? Okay.
Hmm.
Some brittle stars on
Do you
them.
think that's this, or?
What's the genus? Let me see.
Can everybody check their mic? There's
Yeah, someone's mic is rubbing on
Callipertorpha?
something.
Could be.
yeah, whoever just adjusted.
There was
Thank you.
another-
Hey,
Look at that
fish.
fish. You can see
Oh.
through it.
Look at all
No,
these
not
corals here.
Callipertorpha.
Hello, Australia and Moya from New Zealand. The self-sampling crab did in fact make it to the surface.
Callipophora.
It is now
That would be my
a
guess.
specimen.
Yeah, it looks like it.
Hmm.
There's another one on the other page that looks similar.
Are you going down somewhere? Okay. No, you're good.
Were you able to get some stills?
Yeah, did you see this?
are we looking to do any sampling here?
Yes, potentially.
Okay.
Cool.
What is at the base of that?
We saw some of those on the other rocks earlier. My guess would be like a bacterial biofilm, but I'm not sure. Okay. Yeah, no worries then.
Are you gonna move somewhere? Sure, want me to zoom out?
Do you think we can do a sample real quick?
Yes.
Do you want to do like a slurp, a grab?
I'm thinking we'll just do like a snip.
Okay. And then put it into the slurp chamber?
Yeah, we could do that, or
All right.
whatever's easiest for you guys.
Yeah, I think that's, when it's just a little snip, it's easy to cut
Just,
it.
and then slurp it up real
And
quick.
then, yeah, hold it up to the nozzle and
Okay. So
Yeah.
maybe, let's see, maybe the left side on the other side of that anemone looking thing. And we'll just snip that half. So we can leave the brittle star be, let them be.
How big of a piece did you want to sample?
I was thinking the left half of the colony.
over here,
Yeah.
this part.
Mm-hmm.
I love the little nudge.
Very precise movements.
First we have to massage it.
Try to get a few branches.
Gotta make sure it's comfortable.
Make it uncomfortable. That's what it
Standard
looks like.
violence slurp.
All right, so we got another Gorgonian. And then we'll get another Niskin for eDNA
This
as
is
well
sample
afterwards.
036.
Yep. At 425 meters.
Thank you.
Let me see what the genus
Can I
was.
get a ID
Ooh.
for this?
Smooth.
Yes, working on it.
All right. The only slurp jar occupied is 2. Okay, you're going 3?
So do we have to let that
Come what?
go to slurp it?
How's
Yeah,
that?
well, so
Oh, we
they'll
just
hold
hold
it
it,
straight up to the nozzle and slurp as soon as Scott opens the claw. The cutter. Or not.
Testing.
Oh,
there's
there
one of
goes
them.
one.
2, 3.
Four.
Wow. Oh,
I don't see him in the slurp chamber either.
Yeah, just keep it going. Might have to cycle it maybe, I don't know.
So this was a Gorgonian?
Yes, I'm looking right now to try to find.
Where are they?
Let's see.
All of the bolts on the arm look like googly eyes staring at us. What? You never noticed that?
Now I feel like I'm being watched.
Right?
Do they look more like bolts now? I made it a little brighter.
Now they're just blurry eyes. Like, I know that they're bolts.
They're still not coming in the
Yeah.
chamber.
All right, we're gonna do a little trick here.
You gonna flush it?
There's no slurring, yeah. Yeah, suction off when you're doing that. Okay. There you go.
mm.
Oh,
What?
there's
There's
a
bivalve.
There's a skeleton
Another.
stuck in the...
There's
There's a
one.
piece. It's probably just getting stuck up the thing because they're long and thin.
I think there might have been a
2,
claw.
3, 4.
There was like
Hey.
5 or 6, right?
Or
There's
Hey,
5, I
I think
believe.
it was 5.
One more.
Good teamwork.
Is that all of them?
4 of them still.
I think there should be one more.
Was there 2 clippings of it?
Just one that had 5.
Oh, okay. Oh, here we go.
You guys wanted a Niskin bottle, right?
Yes.
Oh, cool. Thank you.
Thanks for
Sorry, I just remembered
Good
that.
job, Scapazi.
Scott Scopazzi.
If you look on the quad view, you're actually looking at the Niskin bottles on the lower left-hand side, which I'm sure a scientist would love to explain, because I will not be able to do a good job.
They're distracted. So a
That's
Niskin
true.
bottle is
Six
a
gorgonian
water
coral
sampling bottle, so
arms.
both
Okay,
ends are
1 second.
open, water can flow through it the entire time, and then and then it is triggered. So Scott is triggering number 9 right now, and as you can see, it'll snap closed after he, maybe?
Yeah, it already snapped.
It did?
it's
Yeah,
close.
The
okay.
third one.
I was looking away.
That's 037.
0,
Sorry, Anthony,
Yes.
I'll explain your question in a second. I'm just writing down notes.
I think
Lily's
Lily
got
was
it.
just explaining it to me.
yeah,
Oh, okay.
You're okay.
Sorry, I'm just totally distracted
it's
right
okay.
now.
You have
okay.
a lot to do when we sample.
Yeah.
So essentially, it just holds a bottle, a quantity of water. I believe this is a question for the scientists, but I believe that these are 2-liter, maybe 3-liter bottles, but they can go up.
I think they might be 5
5 liter?
liters.
Well, so they vary in size and can go all the way up to 32-liter. I think I've seen. So.
can be
But
pretty
essentially, they trigger, and it's a non-pressurized water sample at the depth that it is closed. And then when they take it up to the lab, the water is used how the scientists want, but we will filter out this water with a microfilter, and then we can see what type of biologics and what type of environment is in it.
We have 6 5-liter manually triggered Niskin bottles on Hercules.
There are 5-liter bottles. There we go. Thank you, Brian. But yeah, so
Can we get a still of this? Like
What?
here?
Yeah, I'll grab another one. I just took
Okay.
one.
Is that
Oh,
green
awesome.
thing down there a leaf?
What is that
pink
green
one?
thing?
It does look like
Where
a
is it?
leaf right there underneath this. Let me be the stick of science here. Right there.
stick of science.
Just below the anemone
Oh,
on the right.
yeah.
I zoom in, we can check it out, see what it is.
What did you say your ID for the coral was, Sai?
Callitrophora.
It looks to be a leaf.
It is a leaf.
Not sure the species.
I'm pretty sure.
So green still.
So,
That's a brand new
Scapazi,
leaf.
It
What?
looks
Yeah.
like
what is the
Asiatica.
species of that
Could
leaf?
you pull it up on
I
your
know
computer
it.
Looks like
so
a
I can
hole.
see it?
Yes.
I do not know the species of that leaf.
Ah,
My mom might.
It's a Barringtonia asiatica.
way to go, Nero.
Wow.
Wow.
I'm gonna double check, but I'm pretty sure.
What? Summer's driving.
Yep, pretty certain.
Thank you, Sai. Can you leave that up for a sec?
Oh, you're really gonna write down the leaf
Yes.
species? Sure. Oh, here we go. Go Peyton, go. Doing a great job. So this is my last watch. So I'm bummed, but yeah, it's been fun. We're just currently doing a shift change right now.
Yeah.
So follow along with us. We'll be
All
diving
right,
until
folks.
about midnight. Local
Thanks
time,
for joining
which
us.
is about 8 hours from
12
now.
to 4 watch is about to change over. It was been fun.
Yep.
Hope you guys enjoyed as well. Laters.
Yeah, after this we'll be going
Signing
to
off from
port
the
in Pongo
Benthic
Pongo.
buddies.
You ran that? I ran easy headed. Okay. I did all that stuff.
So we're still off the island of Ta'u in the National Marine Sanctuary of American Samoa, and we are continuing to explore and do some samples, do some fish stand checks, check out some of the geology, and do some
Okay,
photo
signing
mapping in
off.
this
Everyone
area.
have a good afternoon.
So that light glowing you see in the center of your screen are actually our lasers that are 10 centimeters apart, and that allows us to get a sense of size and scale with what we're looking at. Thanks for tuning in from the 12 to 4 watch. The 4 to 8 watch is getting ready to come up right now. And you'll hear most of us again right about 5 as we relieve the 12 to 4 watch to go get their dinner. Excuse me, the 4 to 8 watch to go get their dinner. Brian from the Benthic Buddy signing out.
Hello?
Hello, hello.
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 How's it going, everyone?
Good. Welcome to the 4 to 8 watch.
Bye.
Hey Nav, do we have a waypoint for sample 037?
Yes, we do.
Okay, thank you.
Do you need to look at it?
No, I don't think it's recorded in the event log.
Okay.
So I wanna make sure.
It's a dual sample 036, 037.
Okay, cool, thanks.
Can you fix that for me? Yeah. What bearing has the ship been moving on?
It's been moving 055, but we're going to switch over to 043.
Roger.
Or 040, probably make more sense.
Roger. I don't want to check out this
Yeah, looks fun.
silly thing.
Hello, hello. Yep.
Oh,
Finally, the 4 to 8's back.
Yeah.
Yes.
Our
We're
second
about
dive.
time.
Yeah, it was about time.
Do they have zoom controls for that?
Yeah, somebody does somewhere. I just want to see for a minute. See how it tears?
Yeah.
Pretty bad.
I don't know if it'll work, but
You got controls for this thing?
Whoa. Different perspective on the lasers. I like the lasers underneath.
Come out full wide and do a slow push in if I can.
Are we around 800 meters depth?
422.
Oh, okay. So yeah, deep sea corals don't behave as shallow corals, so they don't need the light. So maybe Georgia can explain a little bit more about that.
Yeah, so one of the key differences is the shallow corals, the reef-building sclerotinian corals, have a symbiotic relationship with a single-celled zooxanthellae. And that lives inside of their tissues, and that's responsible for producing about 90% of that coral's energy through photosynthesis. Down in the deep, obviously we have no light. So they've evolved to not require this relationship, and they're catching all of their food from the water column.
Okay.
What countries are tuning in, Valeria?
Let me check. Oh yeah, we have. Well, the US, Canada,
Ready to move when you are.
Australia, the UK, New Zealand, Vietnam, Turkey, Thailand, Mexico, Jamaica, Indonesia, Hong Kong, Guam, Finland, and China.
Wow.
Wow.
It's pretty impressive.
Yeah, welcome everyone. This is the 4 to 8 watch. Good evening, Jason. It's nice to see you.
You just did gauges, didn't you?
They hadn't been done in a while, so I did it. All right.
We have someone breathing in the mic.
I was just going to say, if everybody could move their mic a tiny bit down.
Welcome, Scotland. That's so great. And Australia.
Problem
for
solved.
Sure. Perfect. The YouTube channel is disabled. Oh, that's a good question.
Looking at your compass.
Oh.
Ready to move when you are.
Okay, I'm good to go.
All right, moving 040.
Roger.
Just going to rotate those suction jars.
Sure.
So how loud is Hercules and Argus? Huh.
Hercules is a fair bit louder than Argus. Say loud?
Mm-hmm.
Yeah, Hercules makes a fair bit of noise, I would think. A lot of vibrating bigger thrusters and all hydraulics. Argus just has 2 small thrusters.
And that for sure scares some fish. Yeah. Do you think the noise scares them more than the light? What do you think, Georgia?
Probably. Yeah, I'd say so.
Yeah.
Yeah, I'd say more of the deep sea guys probably rely less on eyesight.
Yeah, so suddenly having changes in sound and vibrations and things.
Yeah, for sure. Hello, Jim. Nice to see you. Oh yeah, we all hope to see a whale for sure.
Oh, that would be so good.
That would be great.
Keep your fingers crossed.
Yeah. Hello, Portland. I still need to go visit.
That is